Okay, so you had a really nice summary of, 2 years ago we had nothing. Basically, we had 100 million scaffolds, and they were really short, and so on. Suddenly we moved very fast to a position where we now have 21 pseudomolecules, which for you is like, yeah, obvious, right? But for us, it's when we've been working, all my life I worked with ESTs, and suddenly to have a genome browser, it's incredible that you actually have all the genes. Not only that, we have improving gene models, which is really important. We have the A, B, and D genome separate for the first time, so you can ask these questions about expression of the different genomes now for the first time. Before, again, it was just a mixture of the three things all collapsed together. You couldn't see things. Now you can, and you can get that granularity, which is very important because the genomes are not the same. They do have interactions. They do work differently. And it's important that when you try to understand the biology, you understand each one of the genomes and how they interact and so on. So now we have these resources. And one of the other things we started discussing several years ago was how can we actually start getting functional genomic resources in wheat. We haven't talked about transformation, but actually doing wheat transformation is not easy. It's limited to a few varieties. You probably all heard about the NIAB transformation facilities that put out a call for 50 or 100 different transgenes. Many of you actually participated in that, which is great. But it's in Arabidopsis, I'm usually quite envious when you see papers and they have an empty vector transgene. Like, you can't afford to do an empty vector transgene in wheat. It's 12,000 pounds. You basically are trying to scrap for your one transgene, so it's quite difficult. So that's really improving, which is great, but still we said, how can we find ways to make a functional genomic resource that's a little bit easier to do, and it's not just, you have one gene, one shot, cross your fingers that you get it right, which is basically current status, to say, I want to look at 5 genes, what do they do, or 6 genes, and look at them at the same time. And a lot of the basis of biology, in case plant biology, has been this assumption that Arabidopsis, and you learn Arabidopsis and then you can transfer that to crops. We've all said that, right? And now the question is that how true does that actually stand? And I think the challenge now is to say, can we actually do that jump? Can we actually pass some of that knowledge? And Arabidopsis, for us, I worked on wheat all my life, and it's incredible. It's how you can actually get a lot of information and hypothesis from looking at Arabidopsis. You find a gene in wheat, look what it does in Arabidopsis. You have a hypothesis. You can start from somewhere. But I think now is the time to say, can we actually transfer some of that knowledge? And just to put these two things in perspective, if we made the same comparison between Arabidopsis and wheat for humans, basically we'd be looking at a platypus. So basically, this is the comparison we're making. So it's a big jump when we say, we know this in Arabidopsis, therefore it will work like this in wheat. And actually, even for us, when we do an experiment in the glasshouse, it's a huge jump to say that it will work like that in the field. Imagine if you make the jump from Arabidopsis, from the platypus, to say it will work like that in humans. It's a huge jump, and those are things that now, before we had, in a way, the excuse. It was too difficult to work on wheat. Now, in a way, those barriers are being diminished. And it's a challenge for everyone. It's a challenge from the Arabidopsis community to see how they rise to the challenge. It's a big challenge for the wheat community. We've all been trained in QTLs, in genetic markers. We have no idea how to characterize gene. We have very little understanding of really detailed molecular biology because our tools have been very different. So it's about training basically new people who can actually get the best of both worlds and put those things together. And we're generating some of those resources to allow that to happen, and also for us to use. But it's uncomfortable for both sides, just to say that. I mean, from the wheat side, people have said, now all the Arabidopsis people are going to come into wheat. How are we going to compete with them, with their publications, with their profiles, right? So it's uncomfortable for both, but it's a jump we have to make. And in a way, that's why we're here today. We're trying to make some of those bridges and generate some of the resources that are helpful for us, but hopefully helpful for the wider plant community. So what we did is that we made this resource for functional genomics, which is basically just tilling. So if you're not aware with tilling, it's a very simple approach. You take seeds, in this case seeds of wheat, you put them with EMS for 18 hours, you move them around, you grow up these plants, and basically you self them one generation to get M2 plants. And those M2 plants will have lots of mutations across their genome, and those mutations are now quite stable. So in that first generation, they could be a little bit chimeric, but in the second generation, they're quite stable. So what we do is that we collect genomic DNA from that plant, from the M2, and we also collect the seed of that plant, and we archive that seed. So those are two things that we have. And for many years, we used the population gene by gene, and it's fantastic. And it's fantastic because the amount of EMS we can put on wheat is much, much higher than what you can put in a diploid species, because wheat, as having 2 or 3 copies of every gene, will tolerate much higher mutation densities than you can get in a diploid, be it a Rhabdopsis or barley or any other diploid. So tilling populations were developed here in the UK at Rothamsted by Andy Phillips. We developed one a couple in the US, and several labs started doing this because it was really a very powerful way. And just to illustrate that, there's a graph showing the probability of identifying a truncation, so basically a premature stop codon or a change in a splice site. And you can see that because our mutation density in hexaploid and tetraploid wheat is very, very high, actually by screening 1,200 or 1,500 plants, you have an extremely high chance of knocking out a gene, and this is using an average gene of 1.5 kb for the numbers. And so you can have a really, really high chance of knocking it out. If you look at a diploid, and this is the average of many diploid species, basically you would need to screen many, 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 many more. Okay? There's also some drawbacks to this, of course, because many times you have to make double mutants or things like that, and we'll discuss it. But the point is that because of the high EMS dosage wheat can tolerate, it has a much higher mutation rate, so many more mutations per kb, and therefore the number of plants you need to screen is not ridiculous. So we were doing this quite well with PCR, again, working with EST, so getting the A, the B, and the D, making primers specific to the A, PCRing it, and running the dicor gels at that time. And we were thinking, how can we actually take this one step further and make it a little bit more in parallel, because we really wanted to look at more genes than just one gene at a time. And we started thinking about, well, can we actually sequence these mutants? And well, actually, the numbers just don't work. It's just too big. And just to show you, because again, as Nikolai said, most of the wheat genome is repetitive. So you look at Arabidopsis and you see that you have just basically roughly half of it is coding, the other one is repetitive. But then for wheat, roughly 1 or 2% is coding, and the rest is non-coding or unassigned promoter or repetitive. So basically we're looking at a very, very small fraction of the genome, so it would not be feasible to actually sequence all these plants. But then we said, well, what about using exome capture? And the idea with the exome capture is that instead of having to sequence all the genes, or all the genome, we can focus on the coding sequence. It has limitations. We miss all the promoters. But think about it, when we started this, 3 years ago, we didn't have promoters. We had ESTs. We didn't even know about promoters in wheat, basically, unless you actually sequenced a back clone and did a lot of work. So, but as we were progressing with this idea, the technology caught up with us, and it was fantastic, so we could put both things together. So what we did is that we took 1,200 cadenza mutants. So these are hexaploid wheat. It's cadenza is a UK bread wheat variety, and this population was developed by Andy at Rothamsted. And we took another 1,500 Chronos mutants. Chronos is a tetraploid, a durum wheat. Basically has 2 copies of every gene. Cadenza will have 3 copies of every gene. And you'll see how the two are actually quite relatable. And we can take genomic DNA, which we already had. We had already extracted that genomic DNA. And basically we used a nimble gene capture to capture all the exome of wheat. And we did the capture. We basically took those, we put them on a sequencing lane, and then we put them at TGAC, sequenced them, and then used a pipeline established for rice called MAPS to identify those mutations. And basically, once we had those mutations wrapped to a reference genome, we could then say, based on that reference genome, those mutations, if I put this gene model, what is the effect of those mutations on that gene model and on that protein? And again, that was the important thing of sequencing just the exon space, because we wanted to be able to predict what were the mutations and how were they going to affect the protein coding sequence. And then last but not least, we took all those mutations, and because we need to make select for those lines or make combinations, we also, even for us to design primers that will be specific to a genome is not something that's that easy. So basically we developed a program with Ricardo, and basically it's a thing called polymarker, and what we can do is that we can take all those SNPs that we find in the genes, and we can convert them into SNP assays, which in many cases are genome specific, and that just makes life a lot easier when you're trying to make a double mutant or selecting for your mutant out of when you get the seeds. So just to show you a little bit of the internal controls we did to be sure that once we sequenced that was fine. And I just hasten to add that basically I showed this slide here, but that's probably 4 years of a lot of people's work. I'm not going into the details, but it's a lot of people's work at Rothamsted, at the Earlham Institute, TGAC at the time, at JI, at Davis, and so on. So it's a lot of work. I'm just showing this part here, which is kind of the end product. And what we did is that we know that EMS will have a mutation spectrum in wheat of G to A and C to T transitions. That's the spectrum we get. So we wanted to be sure that the mutations we were finding were real mutations. And of course, you can say, well, to have a real mutation, I want to have any position covered by a lot of reads. But as you increase that coverage, you reduce the number of mutations you find because not that many positions in the genome will have really high coverage. So we came up to a compromise where we said, if you look here, this is the number of reads that support the mutation. When you have a heterozygous mutation, when you have 5 reads, roughly 98% of those mutations are EMS-type mutations. So we think we're pretty good. So most likely, if you find a mutant with 5 reads, it's a heterozygous, you're fine. In the case of homozygous mutations, with 3 reads, most of them are EMS type. And of course, if you try to get too greedy and say, if I have 2 reads, it will be a mutation, well, your numbers go down. So the way we presented this was that in the database I'll show you, we presented mutations that have a heterozygous minimum coverage of 5 and a homozygous minimum coverage of 3. Because those mutations, we believe, are 98, 99% true. So when you get the seed, you'll find the mutation 99% of the cases. We also put the lower confidence mutations in the database, so you click a button and you can get the lower confidence that you really are scraping the barrel and you want to find, because basically there's a huge number of mutations in this category, but they're less likely to be real, or still they're 80% or 50%. So if you have the mutation you want, you probably will still order that seed and give it a try. If you look across each line has a variety of, or a variance in the number of mutations. These are all different libraries. And you can see that the majority of the mutations are GA or CT across the different libraries. And the take home, basically the numbers that we came up in the end are these. So we sequenced 1,500 tetraploid lines, 1,200 hexaploid lines. We find roughly 2,700 mutations in Kronos and roughly 5,300 in Cadenza. So that means that in total we have 4.2 million in Kronos and 6.4 million in Cadenza. To put that in perspective, all the exome capture has been done in a huge number of accessions. Basically, all of the SNP database we have are about 1.5 million mutations. So all of a sudden, we create in these 2 varieties basically 10 million mutations in the protein coding sequence of wheat. They'll have many more mutations, but at least in the protein coding sequence of wheat, we can identify that. So this really changes the game in terms of if you want to find a mutant, going to natural variation, forget it. If you can find a mutation here, you have lots and lots of alleles. So we have this database called wheattilling.com. We just submitted a publication for this, so journals are always very picky with that. But basically, we've shared this with anyone who sends me an email. It's a very simple password. We can share that, and it works very simply. And again, these are all the institutions that are involved in this, and it works very easily. You can literally just put in your CSS gene models, so those first gene models that Nikolai mentioned and that Philippa mentioned in terms of having the expression data. We have them all with those old CSS gene models. The TGAC gene models are better. They came out 2 months ago. To do the expression data, Philippa's already done that. It's almost there. And then for us to do this, it would take basically RLM Institute clusters running for probably a year to do all the work we've done again. So we've started, but it's going to take a long, long time to actually get it up to speed. Or you can literally blast it. So you can put your sequence and blast it to find the best hit, because we've also taken all the unassembled reads that we had and we made de novo assembly. So basically, if you have a gene, you can put it in. If you don't have the gene, you have a sequence, you can basically do a blast and you'll find what you want. And literally just put one of the 2 and then you just search. And you get an output, and there's an example of one of the outputs, and I'll just zoom it up here a little bit. So what you get is that it tells you the scaffold it's on, it tells you the chromosome, the line that has a mutation, the category, so it's one of the high category confidence lines, the positions, and then it says a G2A mutation, it's heterozygous, we have 2 wild type and 6 mutant reads. In this case, we have a homozygous 0 to 6. And basically, we have all these SNPs across this gene or across this scaffold in these different positions. And then once we've mapped those positions, then we say, what is the effect of those mutations? And basically, this other half of the screen says, okay, this mutation in this scaffold or this genomic sequence, there's a gene, and basically in this gene, it will be a missense mutation at position 110 of the CDS, P2L, and basically it tells you that there's a missense mutation, for example, or it'll tell you it's a premature stop codon, or it's in a splice junction mutation, or so on. So it'll give you all the categories, and there's a huge list of all the details of what each one of these things means. We have all of that under here, additional resources. You click on that, and there'll be a very well-documented page that will tell you what each column means, what you can refer to, what are the assumptions, and so on. So it's all there. And one last thing here is that we have primers. Again, as I mentioned before, we've taken many of those mutations and we've developed these CASP assays. And with that basically means that you can order that mutant line, order the primers, and then you can go and get your seed and say, okay, great, these are my wild type, these are my mutants, great, I can start studying them straight away. And again, this step might seem pretty trivial, but actually many times you get the seed and then you can't even find the mutation because you have to PCR, specifically the genome you want, sequence, get a mixed chromatogram, start again. Especially people who haven't worked on wheat, it's really frustrating. So we try to make it that we provide primers that they're not going to be the primers that are going to be the most beautiful primers in the world, but these are primers that roughly 80% of the times we test them, we run them, they work 80% of the time straight away without any optimization. So we feel pretty good with that. So in terms of what effects we cause across the genome, so basically these are the number of genes that have at least one mutation. So we basically, in Chronos, we have roughly 48,000 of the genes have at least one mutation, and Condensate 74,000 genes. Genes with at least one truncation, this is a premature stop codon or a splice junction, 60% of the genes have that. Almost all the genes have a missense mutation, and basically 90% of them have either a stop codon or a missense mutation that is predicted to be deleterious based on SIFT. So SIFT is basically, again, it's all documented, but it's a program that tries to look at multiple alignments between different proteins and see how tolerated an amino acid change will be. So not all the mutations will have a significant SIFT value, but some of them do, and basically for most of the genes you can either find a stop codon or that. And then the average number of missense alleles is roughly 22 alleles. So we get, in any given gene, we probably get about 50 mutations, depending on the gene size. Some genes are much bigger, you get 150, 200 mutations across. Roughly 22 of them will be missense mutations, and on average you get 1 or 2 premature stop codons per gene. So that's kind of the stats on that. And one of the things is that, again, knowing a little bit more about your gene model helps a lot, because many times the gene models, because these were the initial gene models, are a little bit fragmented. So actually if you do a little bit of work, you can find a little bit more information. And again, we've documented how you can do that on the website. On that additional information page, we document how you can do it as well. If you have a sequence but not a gene model, how you can actually predict the effect. And perhaps showing a pathway is a good way of exemplifying this. So it's a starch pathway in wheat, and basically each one of these little squares corresponds to, in this case, starch branching enzyme 1. There's 3 versions of that gene, A, B, and D genome. Every time that you have a filled square means that we have a premature stop codon. So this is a knockout of that gene, or that protein, let's say, on one of the mutant that we have. So you can see that most of those squares are filled in. And a lot of the genes that we're not knocking out are basically very small genes, or genes that are probably not predicted very well. And in this case here, we don't have a knockout, but we have a SIFT, significant SIFT of less than 0.05. So for the majority of genes, and you can see now this changes everything, because before you said, well, I don't even know the genes. Now we know the genes. I don't even know the genomes. Now we know the genomes. Where is it expressed? Well, I know where it's expressed now. Well, I want to study it. Well, now you can study it. I don't need to spend a whole PhD getting the mutants. I can actually order the seeds in 20 minutes. So it really changes it. You still need to make crosses, and that's why we made 2 populations, a hexaploid and a tetraploid, and I'll explain that shortly. So the current status is that we have both populations available. Shoot me an email and I'll send you the password, and you can get started. There's no strings attached to anything. It's just that we can't make it public until we get things published. And then access to the mutations. We have mirror collections of seeds at Davis and at JIC, and those are ordered through the JIC seed store. The good thing is that the seed store is completely confidential. It's nothing to do with me or with Andy, the people who kind of developed the research in the UK. It's completely outside of our control. We basically gave the collection to them and they distribute. So there's no information about, you know, who ordered what line. We just know the rough numbers of how many people have ordered, but that's all we know. And we want to keep it like that, because if not, you can get very paranoid about, oh, someone ordered the same line I'm working on, I don't want to know that. There's only 1,200 cadenza lines, so actually there's bound to be people who are working on your same line, but another mutation in that same line. And then the mutants are basically free of the idea will be that we can make them free of IP, so if you find a mutation, you can patent that mutation, you can do whatever you want with it. So there's a £15 non-commercial use fee, so that's basically for research purposes, you can order that line, and a lot of the breeding companies want to have the right to be able to commercialize that in the future, so it's a £200 fee up front. Come back 3 years later, say, oh, I found something really interesting, I want to commercialize it, then you don't deal with me, you deal with the contracts office, and they want to make money. So we negotiated really hard this thing so that it was a relatively small amount up front, but then if you come back later, then you have to deal with them, and there's no misery there. They're going to be brutal about trying to get money. So far, even so, we've just talked about it kind of internally, and to people who've asked, we've had over 800 lines requested from 80 different groups across the world, several breeding companies as well. And actually, we make a comment to know if people are new to wheat, and we get that roughly 25% of the requests we're getting so far are people who have not worked with wheat before. So that's basically you guys, or many of you guys. So there's a resource that's being used already by the Rhodopsis community. In many cases, people who've heard this or that. So it is a resource that's out there. And basically, it's a very simple shopping cart you saw on the web page. I'll just show you here, sorry. Basically, in the web page, it's like this. You click on that little link there, and basically that will take you to this shopping cart. And the only thing you need to do is just put your line name, Cadenza line X or Kronos line, whatever. You say which MTA you want, if you worked with wheat or not, and then you get your seed the next day probably, if you're in the UK. And then the MTAs and all that are there, so you can read the MTAs just to be sure, but the MTA for research is basically, you can use it for research, but if you're going to use it for commercial, please just buy the other one. And one of the things that you have to be mindful about working with wheat is that, wheat is a polyploid. It can tolerate many more mutations when you put high EMS because it can buffer many of those effects. So you'd say, well, do I need to make double mutants or triple mutants to see my effects? And I'll show you there's going to be examples for everything. There's some examples for, let's say, disease resistance genes. You have one gene, you make an effect, and you see a phenotype, very simple diploid-like system. Other things will be a little bit more complicated. And you can go for something like this, with a starch branching enzyme. You can see the wild type, 3 copies, double mutants. They look identical to the wild type, triple mutant, boom, you get the phenotype. But basically you have to make the triple mutant before you see that phenotype. There's other cases, again, it's very similar, the same pathway, but just a gene that does the opposite effect. In this case, a single mutant for the D, the A, or the B, each of the genomes has a 10% effect, so very subtle. The double mutant has a 20% decrease in the amylose. The triple mutant has a 95% decrease in amylose. So again, you'll have dosages from complete, basically you don't see anything, it's complete masking. You might have some dosages, and these dosages might be even 50 or 40, it depends. Okay, and you might have cases where with a single genome knocked out, you have the effect. But it's very hard to know a priori what is it that you're going to get. And that's why we generated these 2 different populations. So many times you will need to create double or triple mutants. The beautiful thing about Kronos, which is this durum wheat, that it's tetraploid, so most likely will have 2 copies of every gene that you're interested in. But tetraploid wheat and hexaploid wheat, for every intent and purposes biologically, they're almost identical. They just diverged 10,000 years ago, so they really are very similar. I haven't seen any study that someone shows one thing happens in tetraploid and it doesn't happen the same in hexaploid. So you're still making a jump, but it's a 10,000 year jump instead of a 200 million year jump. Okay, so for many applications, probably working with tetraploid is a better way to go, especially if you want to show something biologically, because basically you'll see the effect. And you can see here, you can order your mutants. Basically, these plants are spring plants, meaning that they don't need vernalization requirement. You put them, you sow them, and basically 4 months later you have the seed. You can then sow that F1 again, and you can have your F2 seeds and start studying that within a year. Again, you still have to grow the plants. There's no magic, but the resources, at least this discovery, is shortened significantly. And then you have this other case here where you have a triple, where you actually need to make all triple mutant combinations and so on. And again, this is the point why, and I'll just make a plug without any more, I'll just a plug to our paper that we talk about these strategies and so on. You can read more about it there. But the point being that's why I gave so much emphasis to the stop codons or the truncation mutations, because if you have, let's say, 3 missense mutations here and 3 missense mutations there, you need to create 9 double mutants. And then if you don't see an effect, you're still not sure. Does this gene have dysfunction? Or could it be that my missense mutations are just not strong enough to give me the effect? If I have a stop codon in the A, a stop codon in the B, I make a double mutant, and that double mutant is similar to the wild type, then that gene does not have the function that I believe, or there's paralogous or whatever, right? It's a much stronger conclusion that you can reach if you have premature stop codons. So again, in the website that we have, where we have the documentation, we talk about, and you'll see it later in a minute, we have what is the strategy of selecting mutants. Normally we go for stop codons if you want to just determine if that gene is having an effect on that biology, because that way you can reach a conclusion. If you want to get very clever later with amino acid changes, no problem. But many times you want to show that you knock it out and you see an effect. Okay? So these timelines are still, for you guys probably say, well, that's still, still a long time. So one of the things is that we started working with Lee Hickey at Queensland and with Brande Wolf at GI on basically accelerated growth conditions. So Lee started working on extending day lengths to see if we could actually make plants grow faster. And we know we can, but the question was that usually these plants were quite weak and so on. But we started testing different parameters with Lee and with Brande, and we now have basically pretty good established protocols that work in different places now, not just in one chamber in Queensland, but in the chambers there in the glasshouse, several chambers at GI. And basically you get this. This is cadenza. This is the UK wheat variety. This UK wheat can actually be sown in the autumn. It can actually go through the winter without a problem. It was a commercial variety. It works perfectly fine. But actually, this is a typical plant after 38 days when you have it in a normal glasshouse. So 16 hours of light, 18 hours of dark, normal temperatures, 20 degrees, nothing special. That's 38 days. This is in the accelerated growth conditions. So basically, in about 10 weeks, you can go from seed to seed in wheat. And this is not just one variety. This is several varieties. So we've done it for Kronos, for Cadenza, for many other varieties. We've made crosses. We've grown mutants. So all of a sudden now, the whole idea of making this scheme, this is kind of being conservative now. You can actually go much, much faster, and in half a year, get somewhere here, where you can actually have double mutants and start studying them. Okay, so it's really, again, you can see how all of a sudden we have a genome, we have the genes, we have expression, we have the mutants. We can actually grow plants in 10 weeks. So now the barriers are really lowering. So again, I always say to the students, the people who will take the advantage now will be people who actually can, have a mindset perhaps more Arabidopsis-like, that the world is out there to conquer, but can actually work on a system like this, where suppose we've trained many people to be QTL's markers, and we really have to change how we train our people. But at the same time, we really need fresh new blood coming with different ideas from Arabidopsis into wheat and take advantage of the research to really push us as well. But you can see now, and this is not, I'm not showing this to say this is the one picture, I'm just showing one picture. But this is actively working at GI. We have 6 or 7 project leaders who are actually putting their plants through this, and it works perfectly fine. So we're submitting a manuscript with Lee and with Brande, basically outlining the protocols, but it's basically 22 hours of light, 2 hours of dark, nothing special with the temperature, 20-25 degrees depending on the setup, nothing special with relative humidity, very good light levels, that's a key thing. But again, if someone wants a protocol, shoot me an email, and then we can send it to you. I'll copy Brande and Lee so that they know. But basically, we're trying to make this public as soon as possible as well.